Hi everyone, and welcome back to Lecture 1. This is now the second part, which focuses on bacterial anatomy and basically the cell structure and function. Now, a lot of this that we cover in this video should be reviewed from other courses that you've had. So please just send me a message and remind if at any point any of the slides I went over a little too quickly, or if you need a little extra information on anything. Now, before we go into the specifics of the anatomy and infection aspects, the first thing to remind you is the difference between prokaryotes and eukaryotes. So again, always think no. So prokaryotes are any cells that do not have a nucleus and do not have any internal membranes, whereas eukaryotes are like our cells, which have a nucleus and have internal membranes. Now, one of the things I'm going to keep pointing out in today's lecture is to make note that fungi are eukaryotes, okay? So put a little star next to fungi being eukaryotes, because so many times I hear people and students basically refer to fungi such as yeast or mold as if they're bacteria. And that is very, very different from fungi, okay? Because fungi have a nucleus, they're eukaryotes. So in fact, they're more like our cells than they are like bacteria. So please keep in mind that fungi are eukaryotic. So on this slide, we review the domains and kingdoms, and a lot of this, again, this early portion of the slides is more like a review for things you've probably encountered elsewhere. So the three domains, which you kind of saw a little bit of on the last slide, are bacteria, archaea, and eukarya. And when we talk about bacteria and archaea, they are both prokaryotic, whereas eukarya, of course, are eukaryotic. And we just reminded you what exactly prokaryotic means. So again, the idea that bacteria and archaea do not have nuclei and do not have internal membranes. Bacteria is one of the biggest categories that we're going to talk about throughout this semester in this course, whereas archaea we don't encounter as much, but they are still very important to know because archaea are these cells that are capable of surviving in extreme environments. So basically picture these organisms that can live in a volcano or deep within the earth. Okay, these guys are very important to us because if they're capable of surviving these extreme environments, then that means that they have enzymes and proteins that are capable of surviving these extreme conditions. And we've actually isolated some of them to use in research in the medical field. One of the most common ones that you encounter is Taq polymerase. So if you've ever heard of PCR, which we're going to talk about in a later chapter, we're able to perform these experiments with Taq polymerase because we can have this enzyme that withstands very high temperatures because it was isolated from archaea. Now the last category here, the last domain is eukarya. And again, keep in mind eukaryotes, including our cells, have nuclei and they have internal membranes, including various organelles. Now, once again, I want you in this figure over here to put a star and circle fungi as eukaryotic. You can even put a little star underneath eukaryotes here and write fungi such as yeast and mold are eukaryotic, meaning they have a nucleus. So again, fungi falls into eukaryotes, and it tells you that they have a nucleus. Now, as we're considering the anatomy of bacteria and microbes, one of the things you'll have to understand is the various shapes that bacteria can be found in. There are three main shapes that we talk about for bacteria, even though you kind of may sometimes see them listed out in a few more extra shapes. The three main shapes that we always refer to are coccus, which is round bacteria, bacillus, which tells you it is rod-shaped, and spiralia, which is spiral-shaped. Okay, so make sure you know coccus, bacillus, and spiralia. And then the shapes, especially coccus, can be arranged in different ways. So when you gram stain some cells, or you look at some cells throughout this course under the microscope, their arrangement is basically how they line up or how they congregate together. So some of the arrangements we have over here in the corner. You can have diplococcal arrangements, which means a pair of cells. You can have a tetrad arrangement, which is a set of 4. And then you have streptococcus versus staphylococcus. And I want you to put stars next to both streptococcus and staphylococcus, because these will keep coming back throughout the semester, especially since two of the biggest genuses that cause infections in humans are strep and staph. And we're going to kind of study them a lot more in the lab setting. But I want you to remember, streptococcus arrangement, as you can see here, is a linear arrangement. We always remember that as a strip of strep, okay, or strip, meaning a line. Whereas staphylococcus, which comes from Greek for grapes, looks like a cluster of grapes. Okay, so you can think of a staff of nurses clustered together. Okay, so strep is always a linear arrangement, whereas staph is a clustered grape-like arrangement. Now again, we're getting back to the idea of bacterial anatomy. And as I mentioned in the first slide, you have to ask yourself, why study this anatomy of the bacterial cell? So for that, like we said, it helps us understand what to target to kill the bacteria. And it also helps us understand how exactly each type of bacteria is making us sick. So it's very valuable to understand the anatomy of a bacteria. And one of the structures that's going to keep coming up throughout this course is the bacterial cell wall. Okay, so I want you to circle cell wall. Put little stars next to that, because that is a structure that bacteria have, but we do not. And that's going to keep coming back as very important throughout this course. So, as I just mentioned, the most important structure to remember is the bacterial cell wall. Again, this is a structure that bacteria have, but we do not, which is going to come up a lot in this course because that makes it an excellent target for when you're trying to treat a patient. Okay? So when we talk about the bacterial cell wall, we categorize bacterial cells into two different categories based on their cell wall composition. So there are gram-positive cells and there are gram-negative cells. Gram-positive cells, you'll notice, they have a thick layer of peptidoglycan. And what peptidoglycan is, if you remember from one of our previous lectures, that is a carbohydrate structure. Okay, it's carbohydrate with some peptides thrown in as a network. You'll see a picture of it in a slide or two. There's also their plasma membrane, which we also have. Okay, so notice all cells have a plasma membrane. And then for gram-positives, there's teichoic acid sticking out. And we'll talk about that soon with regard to giving certain cells a bit of extra toxicity and also sometimes helping with attachment. Now, what you'll notice when you then look at gram-negative cells is these two cell walls look very different from each other. Gram-negative cells now have a very thin peptidoglycan layer. They then have an extra gap and an outer membrane. And their outer membrane has a lot of lipids and very selective porins. So what I want you to notice is when you look at these two cells, which one do you think is tougher to treat if someone has an infection? Well, which one would it be harder to get antibiotics into or drugs into? Gram-negatives, right? So put a little star over here and write that gram-negatives are not as sensitive to antibiotics and drugs because they have these extra layers and they have very selective porins, okay? Very selective porins. So gram-negatives are more resistant, tougher to treat, whereas gram-positives are more sensitive to antibiotics. The other difference that I want you to know is that gram-positives tend to release what we call exotoxins, whereas gram-negatives tend to release endotoxins. Okay, so we're going to talk about these in more detail at the end of this lecture, but for now know that positives are associated with exotoxins, whereas negatives are associated with endotoxins. Okay, and a little trick to help you remember the difference between the two is I always think to myself, gram-positives are the ones with plenty of peptidoglycan. Okay, that P sound. So this slide is simply to review what I just went over on the previous slide. So first of all, the idea that bacterial cell walls have peptidoglycan, which is a network of sugars and of peptides. You'll see more pictures on the next slide, but this is just one small figure to show you the two sugars that are the main component of peptidoglycan. So they're NAG and NAM. There is a whole lot of this peptidoglycan in gram-positive cell walls, and remember, gram-positives we said are the more sensitive of the two types of cells, and they also tend to be associated with exotoxins, whereas gram-negatives, these guys are the ones with less peptidoglycan but a whole lot of lipids. Okay, put little stars next to that because it's going to be important in later slides as well as in lab, that gram-negatives have a whole lot of lipids. And then put a star next to the fact that gram-negatives are more resistant, so they're going to be tougher to treat, and this is due to those selective pores we mentioned, as well as the extra layers that they have. And the last little point there is a reminder that gram-negatives tend to be associated with endotoxin. Now, after spending so much time pointing out the significance of bacterial cell walls and all of these bacteria that have it, I now want to make note that there is one type of bacteria that does not have a cell wall. And I want you to circle, star, highlight the name Mycoplasma. Okay, Mycoplasma. This is the smallest of bacteria, but what is most significant about it is that these guys do not have a cell wall. An example of mycoplasma is walking pneumonia. Now, it's very important to know that they have no cell wall because now you have to ask yourself, well, why is that significant? What did we say about bacterial anatomy? Knowing bacterial anatomy lets us know how to treat the bacterial infections. And when we do our chapter on antibiotics, you're going to see that a lot of drugs that we give patients, a lot of antibiotics, they target cell walls, including the most famous one, penicillin. If your patient is suffering from walking pneumonia or mycoplasma infection, what's not going to work on them? Anything that targets cell walls, because these bacteria do not have a cell wall. Okay, so for the question, why is that significant that they have no cell wall? You can write, because they cannot be treated by antibiotics such as penicillin that target the cell wall. You must use different antibiotics. Okay, so in addition to having the cell wall, some bacteria may have some other structures outside of the cell wall. So ones to remember are the glycocalyx, fimbriae, pili, flagella, and axial filaments. And all of these you'll hear at various times throughout the course. Now for glycocalyx, there's a few facts that I want you to remember. First of all, the glycocalyx can either be firmly attached to the outside of the cell wall or loosely attached. If it's firmly attached, we call it a capsule. And if it's loosely attached, we call it a slime layer. Now there are three main functions of this glycocalyx. As you can see from the slide, one of them is adherence, so it's nice and sticky to help it adhere, meaning the bacteria can better adhere to your tissues, to your cells, or to surfaces outside of your body. It's also a great protective structure. Okay, so as a capsule or slime layer, it's protecting the bacteria from the outside world, and it protects from desiccation. Now, desiccation, you'll hear again later in the semester, that's basically a fancy way of saying drying out. The third function of a glycocalyx is that, think about it, if you have this structure on the outside of the cell that's basically highly made up of sugar compounds or carbohydrates, during desperate times, if you think of your own body, what you can do with, sugar sources or fat sources, you can start to break them down as a source of nutrients. And that's exactly what bacteria can do with the glycocalyx. So during stressful, desperate times, they can use the glycocalyx for nutrition. Okay, so that's the first structure here on this list. Some cells also have fimbriae or pili, which both help with adherence. Fimbriae are basically look like little hairs, and they will help the bacteria to stick to things. Pili can also be for adherence, but they're also for movement, specifically gene transfer. So during conjugation, which we'll talk about in a different lecture, which is basically cell sex, bacteria can use pili to transfer DNA from one bacteria to another. Now, in addition to the ones that we have for adherence, which are so important because remember, adherence is part of those 5 steps for a pathogen. If you remember, bacteria has to enter, stay in, destroy defenses, do damage, and transmit to a new host, and adherence is basically staying in. In addition to adherence, some structures on the outside of the cell wall are very important for movement. So flagella and axial filament are very valuable to bacteria as they can give the bacteria what we call motility, the ability to move on their own. So flagella, if you've ever seen that in pictures, you'll get to see it on the next slide too. That's basically like having a little propeller. And there's various types of flagella, which you'll go over in lab, that can help the bacteria move on its own. There are also axial filaments, which I want you to remember the term corkscrew. Okay, axial filaments will be on spirochetes that have a corkscrew shape, and the axial filaments not only help them move on their own, but they help them to embed into your tissues as well. So now, in case you missed writing down the notes on glycocalyx, I took a minute to write them all down here so that you can jot them down in your notes. You can pause the video if you need extra time to write them down. Again, just remembering that the glycocalyx can either be a capsule or a slime layer, and that it has 3 main functions, which are allowing it to adhere to things, protecting the cell from desiccation or outside forces, and serving as a source of nutrients in desperate times. Now, to help you visualize some of the structures that we just talked about, you have the glycocalyx over here. So if it's very tight around the cell, it's considered a capsule, whereas loose, it's considered a slime layer. And one of the places that you tend to find bacteria that have a glycocalyx around them is plaque on the teeth and various other biofilms, which we'll talk about that more in other lectures. Biofilms will keep coming back. We also mentioned on this slide fimbriae, which are these yellow structures here that look like a bunch of hairs sticking out from the bacteria's cell wall. You have pili or a pilus over here connecting two bacteria for the movement of DNA from one to the other. You have flagellum, a flagella right here, looks like a little propeller or a whip that the bacteria can spin around to help push the bacteria forward. And you also have the axial filament, which you'll find on spirochetes, that helps them gyrate or corkscrew into things. And an example for the axial filament will be the bacteria, the spirochete, that causes syphilis and allows syphilis to embed within tissues. Now, with some of those structures that we just mentioned, a lot of bacteria are able to gain the ability known as motility, which means that they can move on their own. They can propel themselves in the direction that they need to go. There are three main forms of motility. There's the use of flagella, the use of axial filaments, and gliding motility. So first we're going to look at flagella. When you see trichus at the end of a word, that tells you that that bacteria has flagella. If you're describing the bacteria as monotrichus, lophotrichus, amphitrichus, or peritrichus. Now look at the beginning of the word to figure out what exactly it means regarding that flagellum. With monotrichus, the bacteria has a single flagellum, kind of like you would see on a sperm cell. Lophotrichus, I think of as lots of flagella, okay, lopho, lotso. And the way that you see that is as a ponytail type tuft on one end of that bacteria. Some bacteria instead are amphitrichus, which means they have two flagella, one on each polar end. And then the last option is peritrichus for bacteria such as E. coli. And when you see peri, think perimeter, okay, because those bacteria have flagella all around the outer surface, the outer perimeter, okay? And then no matter which one of these the bacteria uses, whether it's mono, lopho, amphi, or peri, they basically whip them around in order to propel their movement. Another possibility for motility is that some bacteria will have axial filaments, including the example of the bacteria that causes syphilis. Okay, so when you see medical infection example, syphilis, which is a spirochete bacteria causes that, they move around with axial filaments. Sometimes you hear them called endoflagella because they look like a flagella that's been wrapped around within the outer structure of that bacteria. Okay, so it's right under their outer structure. With axial filaments, the bacteria that use that will gyrate in kind of this spiral motion in order to get around, and they'll be able to corkscrew around, which is important in that for some of them, like the bacteria causing syphilis, it also allows them to embed themselves into your tissue, into your organs, okay, by doing that corkscrew movement with the axial filaments. The last type of motility is gliding motility, and there's not as much known about this type of motility at the moment, But basically, these type of bacteria will secrete certain proteins that are like slime-like in order to glide across them. It's kind of like if you've ever seen slip and slides, where you have all of that water and you run and you slide across. That's what gliding motility then does. Now, whenever we talk about the cell wall or cell structures, if you were to just take a bacteria and plop it on a slide and try and look at it under a microscope, you wouldn't be able to see anything. You need to stain the bacteria to better visualize it and to help distinguish the various structures or characteristics of that bacteria. And you're going to get to do the staining in lab, of course, but this is to give you some background information. So there are two categories when it comes to stains. There are simple stains and there are differential stains. When we talk about simple stains, a simple stain is simple because it is only one dye. So for instance, if you just have methylene blue and all of your bacterial cells on a slide turn blue, that's a simple stain. Now, since you're filling all of the bacteria with the same color, you can't tell too many things from a simple stain. All you can tell is size, shape, and arrangement. If you want more information on that bacteria cell or to be able to distinguish things, then you need a differential stain. And what a differential stain is exactly what it sounds like in its name, different colors. You now have two or more dyes, which means now you can distinguish between different structures or different organisms, which is very valuable, especially in the medical field. Some examples of differential stains are the gram stain, the zeolnisen acid-fast stain, and a negative stain. The one that you'll be doing in lab soon is the gram stain, and the gram stain allows us to differentiate cells based on cell wall composition. So basically, is the cell gram positive or is the cell gram negative? Now, if you look at the figures over here, this figure here is an example of a gram stain. The purple cells are gram positive, whereas the pinkish looking cells, those are gram negative. And when you look at this picture, you can ask yourself, what shape are the gram positive cells? Well, they look round, so they are coccus shaped. Whereas the gram-negative cells, what shape are those? They are bacillus, okay, rod-shaped. Okay, so always practice some of the materials that we covered in previous slides. The second example of a differential stain here is the zeolnisen acid-fast stain. And I want you to put a star next to that and write down TB, as in tuberculosis, and leprosy. So the zeolnisen acid-fast stain, in that stain, if a cell becomes red, which I know in this picture it doesn't quite look red, but if it's red, kind of like coughing up blood or blood of limbs coming off, that tells you it's positive as a mycobacterium, which would be either TB or leprosy. Okay? And they're called mycobacterium because they have mycolic acid in their walls. Please do not get them mixed up with mycoplasma, which have no cell wall. Okay? And then we have the negative stains, which are the capsule stains. And in this stain, instead of staining the cells right away, you stain the background of the slide, and then you put a second stain inside the bacteria so you can see which bacteria have a capsule because they have that halo around them. All of these stains, though, you'll notice you're able to distinguish or differentiate either different cells or different structures. So in case you had any difficulty writing down what I was talking about on the last slide with regard to the acid-fast stain, here are some notes I wrote down for you to jot down. You can pause the video in case you need a little more time to write them down. It's basically just summarizing the idea of an acid-fast stain, demonstrating if a mycobacteria such as TB or leprosy is present. Red cells are a positive, meaning yes, the TB or leprosy is present. Blue cells are a negative. And like I said, make sure you do not mix these up with mycoplasma, even though they sound very similar. Okay, so one of the most important stains we have in microbiology, and like I said, you perform this in the BI 219 lab, is the Gram stain. And the Gram stain allows us to distinguish cells based on their cell wall composition, meaning are they Gram positive with plenty of peptidoglycan and very few lipids, or are they Gram negative with all of those extra layers, thin peptidoglycan, and a whole lot of lipids. So to the Gram stain, when you perform this differential experiment, you have 4 main reagents, and I want you to know the purpose for each one and how any mistakes with them would then change your results. So first, you add a crystal violet primary stain. And when you hear primary stain, that means it's the first dye that you're adding to your cells. What happens here is all cells take in the primary stain, meaning both Gram positive and Gram negative will take in whatever the primary stain is that you use. If you use crystal violet, then all cells at this stage will be purple. So Gram positive and Gram negative after the primary stain of crystal violet are all purple. Keep in mind, some people may not use crystal violet, so whatever your primary stain color is, that's what all the cells become. You then add an iodine mordant, and what a mordant does is it complexes with the primary stain. And what that means is it's now making that stain much bulkier to help trap it within the cell. So it helps keep that color in the cell so that it's not easily leaking out. You then add 95% alcohol wash, which is a lipid solvent. I want you to put stars or circle or highlight that fact that the alcohol is a lipid solvent, because where did you hear lipids? Which cells? The Gram negatives. Only the Gram negatives have a ton of lipids, and if it's a lipid solvent, that means alcohol will dissolve lipids, which means that it's starting to break down the Gram negatives cell wall. Okay, so if it's breaking down the Gram negative cell wall, what happens? The primary stain color leaks out now, but only from the Gram negatives. So at this step, what do your cells look like? Gram positive is still purple, or whatever your primary stain color is, whereas Gram negatives have lost their color. They are colorless. It leaked out. You then add the final reagent, which is the counter stain. In this example, it's safranin, which is a pink color. The counter stain can only enter any cell that's currently colorless, because think about it. If you try and put the counter stain in those Gram positives, they're full of dye already. There's no room for a counter stain. So instead, the counter stain will only enter empty cells. Which cells are currently empty or without a stain right now? The Gram negatives. So at the end of this stain, the Gram positives still have the primary stain, whatever color that was, which in this example is purple, and the Gram negatives now have the pink color, the safranin. Now you have to ask yourself, what are some things that can go wrong, and what would the cells look like if any of these errors happen? This you'll have to practice in lab and in lecture. So on the next slide, I'll give you a little practice. So here are some examples of what I was talking about when I said gram stain problems. Okay, so when you are kind of studying gram stains, to determine if you truly know the meaning of the gram stain steps and the function of each reagent, you should be able to troubleshoot if any of those reagents were forgotten or swapped. Here are just two examples, but there are many other ways that I can ask these. The first one says, if a student accidentally swapped the crystal violet and the safranin, what would happen? So that basically means that they did safranin as the primary, then iodine, alcohol, and crystal violet. So whenever you do any of these problems, go step by step. Okay, there are 4 steps that you should normally be seeing, and ask yourself what happens after each of the steps now. So if they first added safranin, which is a pink color, what happens with the primary stain? Well, it goes into all cells, including the gram positives and the gram negatives. So all cells would be pink at the beginning. Iodine would then help trap that color in the cells. Alcohol would then do what? Dissolve the lipids of the gram negatives. So now the gram negatives will lose the pink color, and what would enter them? The crystal violet. So in this scenario, now the gram negatives would be purple and the gram positives would remain pink. In the second example, we ask, what if they forgot the alcohol step? If they forgot the alcohol step, once again, go step by step to what they're doing and see what would happen. So they remembered the first step, which is normally crystal violet, so all of their cells turn purple. They then add iodine, which is the second step, and that helps trap the purple color in. But now, without the alcohol, what doesn't happen? Well, gram negatives never lose their color, so everyone still has purple inside of them. So what happens when you try to add safranin or a counter stain at the end? Can't enter any of those cells, right? Because all of those cells are still full. So at the end of this gram stain, everyone's cells, both gram positive and gram negative, would be purple. And so this would no longer be a differential stain, but would instead act like a simple stain. If you want any extra practice problems for gram stain issues that can occur, just ask me in Remind or through email, and I'll give you more practice, especially before the next exam, which will cover this material. Now, in addition to the cell wall, there's also the plasma membrane, which is right below the cell wall. Now, keep in mind, I mentioned our cells also have the plasma membrane. So even though we hear it constantly said that the plasma membrane has no pathogenic qualities, but may be a target for therapeutic strategies, make sure to write next to that it's not a good target, that usually we do not want to target the plasma membrane because we can hurt the patient as well. What is important about the plasma membrane is that it greatly affects the permeability of cells, meaning it guides what can enter a bacteria and what can exit a bacteria. It also houses the electron transport chain, meaning that it's part of how bacteria can make their ATP, or their energy. The last thing to note here is that the plasma membrane is very important for transport, and you have to know the difference between osmosis versus diffusion, which are passive and active diffusion. When we talk about these 3 types of transport, the first one is osmosis, and I like this little figure here for osmosis. Okay, that demonstrates that osmosis is the movement of water. Okay, and when the water is moving, it goes based on low solute to high solute, which is a fancy way of saying from high water to low water. Now, when you see that movement of water, okay, if you have a hypotonic solution, hypotonic means low salt, right? Low salt around that cell. So if water is moving from where there is low salt, which is around the outside, then it's going to be moving from the outside to inside the cell, which is why in the figure here you see the cell swells and then can burst. The way that I remember what happens in hypotonic solutions is I think hippo, hypo. So whenever you hear hypo, think hippo, getting really, really big. The cell gets really, really big, it swells and can burst. The opposite is true in hypertonic solutions. So hypertonic solutions, which is this guy here, if it's hypertonic, that means there's a lot of salt on the outside of the cell. Whereas in comparison, the inside of the cell is lower salt. And as we just said, the water moves from low salt to high salt. So in this case, it's exiting the cell. Okay, so the cell shrivels. To help you remember what happens in hypertonic solutions, always think of salty environments, right? If you eat a ton of salt, what happens? Your mouth is all dried out, right? You get that drying out, kind of like the shriveling cell. Or if you've ever heard of what happens when kids pour salt on slugs, they shrivel up. Okay, so you get the loss of water. The ideal, which for all of our nursing students or anyone going in the medical field, is isotonic. Isotonic means there's a balance inside and outside the cell. So the water is not going to be lost or gained in any high amount, which is what you want because you don't want cells bursting or shriveling up. So you'll notice in hospitals, a lot of the solutions that you use will say isotonic buffers. The other types of movement that you can have from a cell's plasma membrane is either active transport or passive transport. When you think about these terms, always ask yourself, what does active mean? What does passive mean? So if something is active, that means it's high energy, right? So active transport requires a lot of energy, which means that it's movement against how cells normally want to move things. So if cells normally want to go from high to low, right, because they want to equal things out, active transport will move things from low to high gradients. Okay, so low solute to high solute. And this now, instead of the water moving, this is the salt or the solute themselves moving. Passive transport, on the other hand, is passive because it doesn't require energy. It's exactly the movement in the direction that things normally want to go, which is from high to low. Okay, because high to low would then balance things out. Okay? So keep in mind, again, osmosis, what happens to a cell in each of the three conditions. Make sure you know that. And know the difference between active and passive transport. So the idea that active transport requires energy, which is because it's going against the normal concentration gradient, because now it's moving things from low to high, versus passive transport, which does not require any energy because it's happily going in the proper gradient direction of high to low. So the last of the structures outside of the cell wall that we'll talk about before moving on to internal structures are endospores. I like to call endospores little escape pods for certain bacteria. Now keep in mind, only certain bacteria are capable of forming endospores, but the ones that do, these endospores make them very dangerous to us. So what an endospore is, you can see it in this figure here. It's basically a protective structure. So these certain bacteria, what they can do is they can undergo sporulation, meaning if the environment around them is suddenly very dangerous, they can protect themselves from it by leaving their vegetative active state and instead forming an endospore that is a double capsulated structure with just DNA inside. Okay? This protects them from the outside environment, which at the moment is very unfavorable to them, either due to things like very high heat or toxic chemicals or UV, etc. So in this protective endospore, they are now highly, highly resistant. What they then do is they stay in this state of dormancy, meaning inactive, while the threat is there. And then when conditions start to get more favorable, for instance, if the temperature has now dropped to a very comfortable temperature, or if the toxic chemicals have gone away, what they then do is they germinate. And what germination means, just kind of like picturing a bulb coming out from the ground as a beautiful flower, what these guys do for germination is their outer capsules of the endospore will crack and allow water in, and the cell will then return to its nice big vegetative active state because conditions are better. Okay? Now, keep in mind, I mentioned before that endospores are very dangerous to us because of the fact that they're so highly resistant to all of the things that we usually use to try and kill bacteria. So things like high heat, desiccation, which we said before is drying out, toxic chemicals, antibiotics, UV, and even boiling. If a bacteria can produce endospores, they can survive all of this. The big clinical example to remember is botulism, which we'll talk more about in later chapters. But botulism, for instance, most people associate with canned products, and the reason why botulism survives the canning process is because it can go into endospores and stay dormant until the canning process is over, and that can is now sitting on a nice, comfortable, perfect temperature, perfect environment shelf in your house or in the store. Okay? So again, we'll talk more about this in later chapters, but for now, I want you to remember whenever you see endospores, think extremely resistant. Remember sporulation, meaning they form endospores when the environment is stressful. They are dormant, and then they germinate when the environment is better. Okay? And keep remembering that example, botulism. So that's it for the outside anatomy terms. Now we're going to go through some internal anatomy structures of bacteria. The first internal structure to remember is cytoplasm, and we also have cytoplasm. There's two terms that you'll hear associated with this. Sometimes you hear the word cytoplasm, whereas sometimes you hear the word cytosol. The difference is that cytosol is simply the fluid within the cell, whereas cytoplasm is the fluid plus all of those dissolved suspended particles such as proteins, carbohydrates, lipids, and ions. Okay, so when I wrote here, this first bullet point here, this is the definition of cytoplasm, whereas cytosol would just be the fluid. The other term here to know is cytoskeleton, and the cytoskeleton, just like our skeleton, is for keeping shape and structure. So the cytoskeleton is made up of tubulin-like proteins that help keep the bacteria looking like a bacteria. Another internal structure of bacteria is the nucleoid, because remember, what did we say about bacteria? They're prokaryotes. No means that they do not have a nucleus. Instead, they have a nucleoid, which will resemble the nucleus, meaning it has DNA in it, but it has no nuclear membrane. Within the nucleoid, you'll find the bacterial chromosome, which is a big circular ring of DNA that has all of the cell's genes that are critical to its life functions. Bacteria also have what are called plasmids. So keep in mind, plasmids are extra chromosomal, meaning that they're beyond the regular chromosome, which means not all bacteria will have them, and the bacteria that do have them they have various different versions of plasmids. What's important about plasmids is even though they're not essential for the life functions of that bacteria, because we said they're extra, just like some people I know, what they can do is they can confer an advantage to the bacteria. And the big advantage that they can give that I want you to remember is antibiotic resistance. So when we talk about antibiotic resistance in lab and in a later lecture, remember that bacteria get antibiotic resistance from plasmids, from genes on plasmids. Okay, so when I wrote what may be an advantage to a bacteria, the answer is antibiotic resistance. Then there are ribosomes, and ribosomes you actually find in both prokaryotes as well as eukaryotes. Now, whenever you hear the word ribosomes, I want you to immediately think of the function to make proteins. And they do this, they make proteins by translation, okay, converting an RNA blueprint into the necessary amino acid chains. Now, with ribosomes, if you are looking at a prokaryote, such as bacteria, you will find them free floating in the cytoplasm. Whereas if it's in a eukaryote, such as our cells, we have organelles, you'll find a lot of the ribosomes actually attached to what's called the rough ER, the rough endoplasmic reticulum. And it's called rough because it's coated in all of those little ribosomes, which you'll see a few slides from now. The last of the structures to know are inclusions, and I always tease the students saying, oh yeah, you have to memorize every single one of the ones on this slide, but that is a complete lie. All I want you to know is that the term inclusions means structures that hold things. All of the bullet points on this slide are just examples of inclusions, and as you can see, each one can hold different things. So some of them are holding phosphates, some are holding energy, some are holding various enzymes or proteins. But ultimately, the thing to remember is inclusions are simply little sacs that hold things. Now let's focus on eukaryotes for a little bit because eukaryotes and eukaryotic cells are important since some microbes such as fungi are eukaryotic, as well as you who end up being the host to a lot of the microbes. Now we're going to go through some of the organelles we mentioned, what they actually mean. The first one is the nucleus, and you can think of that as the brain or the command center of the cell, because that is the organelle that will contain the genetic material of that cell. And in eukaryotes, this genetic material is in the form of chromatin. And what chromatin is basically, think about it. If you have DNA, a DNA strand is ridiculously long. Like if you unraveled it, it would be around like 6 feet long. And so how are you going to fit that in a tiny microscopic cell? Well, what the cell does is it takes that DNA and it wraps it around proteins so that now it's a nice, compact, squished up ball of DNA that can fit inside the nucleus. And there'll also be some RNA mixed in as well to help maintain that shape. Okay, so the DNA and the genetic material of that cell is found in the nucleus. Now, earlier we mentioned that there is the endomembrane system. What the endomembrane system is the compartmentalization of the cell, meaning picture endomembrane means inside membranes. So all of those membranes helping to separate the different structures in the cell. So that includes the nuclear envelope, the endoplasmic reticulum, the Golgi, and the vesicles. Now we've kind of already mentioned what the nucleus does. So the nuclear envelope helps hold in all of that DNA and genetic material. Next, we're going to talk about the endoplasmic reticulum. Now, some people think of the ER, or the endoplasmic reticulum, as the roadways of the cell. And that's because there's basically a whole lot of transport going on in these roadways. There's some protein transport and some lipid transport. Now, we've already mentioned the rough ER, and I said that it was rough because it has all of those ribosomes stuck to the outside of it, right? So it's lined with a whole bunch of ribosomes that make it look rough, and you could think R, rough ribosome. Now, as soon as you hear that the rough ER has a whole bunch of ribosomes, what does that tell you its function is? The rough ER can produce and modify proteins. Okay, so when you think of the rough ER, think of ribosomes and think of protein making and transport. And it also modifies the proteins as well, meaning it can add extra, extra element groups to them. So for instance, it might phosphorylate them, stick on a different group. Okay. There's also the smooth ER, and when you think smooth, well, it's smooth because it does not have ribosomes on it. And when you think smooth, I want you to think waxy, oily, right? Oily smooth. Well, that's because the smooth ER can produce lipids. So that will help you remember. If you think smooth, even though they're smooth because they don't have ribosomes, when you hear smooth, if you think oily, waxy, it'll remind you that the function of the smooth ER is to produce lipids, which can include fat molecules and hormones. And then what they do, the reason why it's thought of as roads or streets, is they package up these molecules. So they form vesicles, meaning little round sacs, or picture a round bag. So they're bagging up these lipids that they just produce, and they'll transport them in vesicles to other parts of the cell where they are needed. Okay, so ER, or the endoplasmic reticulum, helps produce molecules and also helps package and ship them out. Now, the big packaging plant, however, is not the endoplasmic reticulum. The big packaging plant is actually the Golgi apparatus. And what's important about the Golgi apparatus is it will take in those vesicles that came from the endoplasmic reticulum. So if you look at the figure on this slide, you have the nucleus here. This is the rough ER because you see all of those speckles of ribosomes. Then you have the smooth ER, and inside the smooth ER are all those lipids that it's been producing and modifying. Now notice from the endoplasmic reticulum, there'll be these little transport vesicles that then enter the Golgi. This big structure here is the Golgi apparatus. So vesicles containing the produced lipids or the produced proteins from the endoplasmic reticulum then enter the Golgi apparatus, and in the Golgi apparatus, they then will modify those proteins and lipids, so they will change their structure a bit. They might add on certain molecules to them, and then they package them in new vesicles to then ship completely out of the cell or ship to other parts of the cell if needed. Now, if they are shipping a vesicle containing these modified proteins and lipids completely outside of the cell, we call that secretion. The cell has secreted those substances. And the way that they do that is the vesicle, you'll see in this picture, it merges with the cell's membrane and then spits out its contents. Okay, so remember that the ER was producing the proteins and the lipids and putting them in a vesicle to ship to the Golgi, which then modified them further and shipped them out of the cell or to a different part of the cell. Now, the Golgi will produce some very important vesicles that are not just, modified lipid or protein. to move elsewhere, some of the Golgi vesicles produced are what we call lysosomes. And this happens in animal cells, not in plant cells. And what these lysosomes are is a vesicle or a storage sac full of very powerful digestive enzymes. And that'll be very important in the cell because the cell will then use lysosomes to help digest its materials that it needs to break down. In fact, it will even use the lysosomes to destroy any damaged organelles or damaged proteins that might cause problems within the cell. So it ends up using them as a basically recycling plant or a garbage plant. And even the lysosomes, scary enough, they're even involved in programmed cell death. So for instance, sometimes cells that will get infected, they basically know that they are a problem for the overall well-being of that cell or organism. So they will have programmed cell death where the lysosome will engulf and break down that cell, okay? Or release the digestive enzymes in it to destroy the cell. Now, if you think of the lysosomes, I want you to think of them as a big ball of digestive enzymes. They help break things down in the cell. Now, in addition to the endomembrane system, which included those vesicles that we were just talking about, even the central vacuole and peroxisomes, we also have the energy-related organelles. And like I said, the energy-related organelles are mitochondria, and in plants, it includes chloroplasts. Now, when you think of chloroplasts, again, think of these as being in plant cells, not animal cells. They are a double membrane enclosed organelle, and they use what's called chlorophyll to help capture sunlight. And the chlorophyll you find in what we call thylakoids. So when you see these little structures here that look kind of like a Mento, these little structures here are thylakoids stacked on top of each other. And those thylakoids have chlorophyll, which helps capture sunlight. And that's critical in things like plants, algae, and even certain bacteria that use photosynthesis. Okay, so chlorophyll you'll only find in things like plants, algae, bacteria, and the function is for photosynthesis, for turning sunlight into usable energy in the cell. Now, when you think of these, again, keep in mind you're thinking of cells that can produce basically their own food. They're producing food, they're producing energy for themselves. Okay, energy-related organelles. There's also the mitochondria, which you always hear called the powerhouse of cells, and powerhouse because it is the big energy maker of the cells. What they do is they carry out cellular respiration, which if you visualize the formula for that, you'll see in later chapters, it's actually the opposite of photosynthesis. So in photosynthesis, the plant cells or the cyanobacteria, they're making glucose, they're making sugar molecules, basically their food, and also energy. In the case of mitochondria and respiration, what's happening is glucose is being broken down. So instead of making glucose, they're breaking it down to get more energy. Now, the way that you see this happen is they will make carbon dioxide, water, and energy from carbohydrate, specifically glucose, using also oxygen. Now, when we talk about mitochondria, you'll see them as organelles that have two membranes. They have an inner and an outer membrane, and that will include this semi-fluid matrix with DNA and ribosomes in it. And so as soon as you see that, you see that they're capable of making proteins, right? Whenever you see the word ribosomes. And so mitochondrial DNA and proteins, they're basically encoding for and producing proteins that are involved in what's called electron transport. And also, sorry for the big scary term, you don't have to memorize it, also oxidative phosphorylation. And that's a fancy way of saying making ATP. Okay, that's involved in making ATP and energy. So you don't have to memorize those terms, just know that the DNA and ribosomes that you find in mitochondria, sometimes it's called mitochondrial DNA, they are used for making proteins involved in electron transport and ATP synthesis. Okay, that's why they're so important. Now we mentioned that there's also a cytoskeleton in cells, kind of like your own skeleton. It'll be made up of a whole bunch of protein fibers kind of stuck together and having motility. And these are very important because they have two main functions. They're involved in maintaining the shape of the cells, and also they allow for movement of the cells themselves, as well as movement of the organelles in the cell. Okay, so again, when you think of cytoskeleton, cyto means cell. Skeleton is just like your skeleton. It's maintaining shape and allowing for movement. Okay, that's a wrap. So you can take a break now and then move on to the last part of Lecture 1, which is Part 3, focusing on metabolism. Again, if you have any questions, anything you need help with, or you want some practice problems on anything, just please send me a message in Remind.